thank you very much. And thank you also for being so patient, especially to François-Joseph Lapointe, who is our next speaker. François-Joseph, hi, and thank you for sticking around. François-Joseph is a scientist and bioartist. He obtained a PhD in biological sciences from University of Montréal and a PhD from University du Québec à Montréal, for which he developed an algorithm that designs dance choreographies based on DNA sequences. Preferring the term paradisciplinary to interdisciplinary, François-Joseph has developed two careers in parallel. His bioartistic practices have kept him busy with subjects such as the question of self in the microbiome era and how to use metagenomics tools to do art. And I present you Jean-François Lapointe. François-Joseph, sorry. Salut.
Okay, so I will share my screen. Can you hear me?
Yeah.
Yes, we can hear you.
Okay, I'll share my screen. Go to PowerPoint. Okay. Is it working?
Yes, it's working. We see a PowerPoint.
Can you see me as well?
Yes, we can see you in the margin with all the other speakers.
Okay, I will turn off my video if you don't mind.
I don't know. You just turned off your shared screen.
Yeah, yep, because I cannot see. Okay. There you go. So is it working now?
Yep, you've got your full screen.
Okay, so hello, and thanks for having me. 50% of the cells in your body are not human cells. 99% of the genes in your body are not human genes. You are not what you think you are. You are what you breathe. You are what you eat. You are what you digest. You are what you shit. You are what you touch. You are what you fuck. You are what you pray for. You are what you're born as. Everything you do changes what you are. Everyone you meet changes who you are. You are not human anymore. You are a superorganism. You are the sum of your genome and the genomes of every bacteria and viruses and fungi and other microorganisms living on you, around you, and inside of you. You are a walking bag of microbes. You are your microbiome. I am my microbiome. And as a biologist, I am interested in the human microbiome to quantify the diversity of microbial communities living on human bodies. As a bioartist, however, I have a deep interest in the human microbiome for totally different reasons. I wonder about the limits of the human species living in that sea of microbes. Namely, I'd like to assess whether my microbiome is affected by my behavior. Is my microbiome changing in response to the people I meet? How is my microbiome affected by my interactions with the outside world? To do so, I create experimental performances in which I use my body as a canvas to study the transformation of my own microbiome. And today, I will focus on 5 series of experimental performances I've been doing over the last few years. First, you are what you touch. It's the famous 1000 Handshakes project I set up to map the microbes between us. This is a very strict research protocol that I'm using, not unlike what scientists are using. So my artistic performances rely also on the research protocol. In the present case, that involves going to a new city, shake hands with as many people as possible, at least 1,000. Count the number of handshakes, collect skin microbiome at regular intervals, that's every 50 handshakes, sequence the microbiome samples when I get back to my lab, and generate what I call microbiome selfies. So this was Paris a few months ago. Let's see if you can hear the sound. 651, 652, vous pouvez être numéro 653. Can you hear the sound?
Yes, we can hear. It's quiet, but we can hear.
660, 661, and you are number 689. Good for you. 717, 718. No, I need to reach 1,000. Partez pas sans me serrer la main. 730, 731, 732, 733, 734, 735, 736, 737, 738, 739. Le problème, ouais. Bonjour. Bonjour. I'm counting handshakes, and I'm trying to reach 1,000. This is 790, 791. Hello. 792. Vous parlez français? Je dois serrer la main à 1000 personnes, vous êtes numéro 800. 805. I'm counting handshakes. 841, 842. Je pense que c'est près de la fin, là. Bonjour. Je vous ai déjà serré la main. 851 et 852. Je dois serrer la main à 1000 personnes aujourd'hui. Je vais être le numéro 892. Non, mais je peux ne pas tenir compte de votre score, mais quand même vous serrer la main.
Je suis occupée, monsieur.
D'accord, je m'excuse.
Je vous souhaite une très bonne journée.
Vous aussi. 67e. 968. 973. 974. Bonjour, monsieur. Serre la main à 1000 personnes. Vous êtes la 975e personne. 996. 997. 998. 999. 1,000. Voilà. You're the winner. This is you. All of you. Ouais, en 3 heures, 1000 poignées de main. C'est un bon score. Je suis satisfait. Well, the same experiment has been repeated over and over again in different cities. So this was at Culture Night in Copenhagen, 1,218 handshakes. Nuit Blanche, Montréal, 1,001 handshakes. Quantified Self exhibition in San Francisco, once again, over 1,000 handshakes. The National Experimental Art Forum in Perth, Australia, 1,000 handshakes. The Transmediale in Berlin, and you can see people's faces as I'm shaking their hands. Culture as Medium in Baltimore, where I shook hands with a clown. And then I go back to the lab. In each case, the same protocol. Shake hands, count the number of handshakes, sample the microbiome, and then sequence the data and create what I call microbiome selfies. So here is one, a microbiome selfie of the microbiome sampled from my right hand after 150 handshakes. This image can be read as a social network. It's like a Facebook for microbes, in which different bacteria are connected to one another based on their genetic similarities, which allows one to identify clusters of bacteria more or less connected based on their genetic resemblances. 300 handshakes, 500, after 650 handshakes, 850, and 1,001 handshakes. Second project, you are what you eat. Everyone knows that phrase, but is it true? I wanted to test it. That is, trying to change my identity by changing my diet. You'll see why there's a bat on that slide in a few minutes. So here again, I follow a strict research protocol. Go to a new city, identify a local food specialty, prepare the food and eat it as locals would do, collect my oral and gut microbiome before and after. The gut microbiome is usually the next morning, sequence the microbiome samples, and then generate microbiome selfies. This is the Becoming Danish performance I did in 2018 in Copenhagen. For 5 hours, I invited people from the public to select typical Danish dishes at a buffet, then sit down with me to eat the selected dishes. And after each guest, every 5 minutes or so, I sampled my oral microbiome. And as I said, the next morning, my gut microbiome. In 2016, I did the same thing in Baltimore, eating kimchi for 2 hours in order to become cabbage. Yet, the most famous You Are What You Eat performance is the one I did to become Batman. It was in 2014. I was doing a field trip in Guinea with my colleagues from the French Museum of Natural History. And since we're collecting bats and local people were eating these bats, I also ate one bat. Let me repeat, 2014, Guinea. Does that ring a bell? Anyone? We were right in the middle of the Ebola outbreak. In Africa, where bats are suspected to be the reservoir of the virus. By eating this bat, I could have been infected by the disease, but I survived. And back in the lab, I sequenced my oral microbiome and the bat microbiome that we had collected, and I created a microbiome selfie for Batman. In red, it's my microbiome, the human microbiome. In yellow, the bat microbiome that is now part of my bacterial identity, my new Facebook friends. Yet another project, one of my personal favorite, You Are What You Fuck. That one is more tricky because I cannot do it alone. So the first step of the protocol was to find a sexual partner. Have her or him sign the consent form, very important. Then engage in a variety of sexual practices, collect multiple microbiome samples before and after sex, and then next step, over and over again, sequence the microbiome samples and generate microbiome selfies. Well, I was fortunate enough to have a partner ready to perform with me. Thus, we sampled both of our oral microbiome as well as our digital microbiome, that's our fingers, anal microbiome, urethral, and vaginal microbiome. And the rest is what I call microbiome Kama Sutra, a combinatorial game in which the microbiome samples are compared before and after different sexual practices. If you look closely, you'll notice that some combinations were not tested, either because they were anatomically impossible or because they were rejected by one of the participants. Let me show you just one safe for work microbiome selfie. This one is entitled The Kiss. It represents the coming together of my oral microbiome with the oral microbiome of my partner after a long French kiss. I'm currently working on this other project, the Holy Microbiome. Once again, the research protocol involves going to a new city, identify a religious or holy site, engage in a ritualistic practice as pilgrims would do, collect the microbiome samples before and after the ritual, sequence the samples, and generate microbiome selfies. This is me in Vatican City 2 years ago during the Jubilee. I licked the holy door at St. Peter's Basilica, drank holy water, and swabbed my tongue to collect a microbiome sample. This is me again in Varanasi, India. I put my feet in the Ganges River and then collected a skin microbiome sample. The Wailing Wall in Jerusalem is another holy site I visited, swabbing my forehead before and after. And more recently, I was at the Blue Mosque in Istanbul, all of these samples are currently being processed. So no more microbiome selfies to show at present time. Last but not least, let me tell you about this microbiome rebirth incubator I've been working on with Marianne Cloutier, a research assistant in my lab. This art installation was on display at the Science Gallery in London. That's London, UK, not Ontario, as well as at Ars Electronica in Austria a few years ago. It is a piece that addresses the worldwide cesarean section epidemics. The impact of delivery mode has been at the heart of a heated debate in the microbiome literature on the effect of C-section on future health of babies. It is known that newborns delivered by C-sections are more likely to become obese as adults and to develop autoimmune and allergic diseases like asthma. For that reason, the practice of vaginal seeding or vaginal microbiome transplant has been proposed to circumvent the negative effects of C-section. Basically, it is quite simple to do in cases of planned C-section. Just need to put a sterile cloth in the mother's vagina prior to birth, and then wipe the baby's mouth, face, and body with it after birth. Although the benefits of this medical procedure are currently debated in the scientific literature, it also has one important shortcoming. It doesn't apply to emergency C-sections when time is a factor to save the baby's life or the mother's life. So this project has a totally different research protocol. I would need to find a breastfeeding mother, collect a breast milk sample, collect a vaginal microbiome sample from the same woman, pour milk and microbiome sample in one giant incubator, shake well, and then seed the baby with this probiotic cocktail. The microbiome rebirth incubator, and you see a performance that was presented at Ars Electronica. Those are some of the pictures that we took alongside the installation of the incubator. So it's a piece that collapses together the moment in birth and that of breastfeeding, when different strains of bacteria are transferred to the baby to construct the immunity system. By mixing together in this giant incubator a vaginal microbiome sample and a breast milk sample, we are creating a probiotic cocktail for babies born by C-section, or baby kombucha, as I call it. Of course, the piece is not about building an actual incubator. It's a metaphorical piece which addresses the physical, psychological, and symbolical trauma of having a C-section. And you can see here the piece installed at Science Gallery in London. And I'll end with that. Thank you for your attention. And if you have any questions, I'll be happy to answer. And I will turn on my camera right now.
Wow, thank you so much, François-Joseph. That it's such a privilege to have you here with us and see examples of your work, which is just, they're so extraordinary and thought-provoking but also delightful. You sort of managed to really hit that balance between like making us think and creating these beautiful aesthetic works. So I think we have a lot of comments and questions in the chat. So Kat, why don't you let us know?
Absolutely. So there's two main questions so far. The first one, do you have any idea how long these microbiome changes last after your experiment?
That's a very good point, because since I'm only collecting one sample after I'm finishing the experiment, I really don't know. But there's been many, many scientific studies about the long-lasting effects of microbiome changes. So depending on what you do with your body, like your hands, now we're getting used of washing our hands very often nowadays. So the microbiome will change very, very rapidly. But in other areas, in other parts of our body, then the microbiome will probably remain more or less the same. We have what we call a core microbiome, which is a group of bacteria which more or less stays with us and sticks with us and can be used as a way to identify who you are on crime scenes. And there's like these satellites, microbes that are changing all the time.
Wow, very interesting. Sort of on that note, you're talking about the hand washing. How will this current kind of pandemic affect how you proceed in the future with? Do you have any plans about going forward with this kind of thing?
Well, I was supposed to be in China a couple of weeks ago to actually do the 1,000 handshakes. So of course it was postponed. So I'm currently trying to figure out a way to do this in a safe manner. So like we had safe sex after the HIV pandemics, maybe we're gonna have safe shapes after the COVID-19. So, so I think it's really challenging for me, because I would never have guessed that the most dangerous performance I would set up is not eating a bat, but shaking hands with people.
Right. Yeah.
So, yeah, it will change everything.
Wow. A couple other questions here. Do you think that the effects on this practice about over our microbiome could also affect our immune systems?
Oh yeah, for sure. For sure. There's a direct relationship between immunity and the microbiome. And that's why C-section babies have a different immunity system than babies born naturally through the birth canal. We're working on the effect of immunity on not simply on human species, but we're looking at bats at the moment. And what is the effect of using different types of bacteria to boost the immune system in bats. So of course, it is well known that the microbiome is totally related to our immunity system.
I think it's also known, or at least there's research that's combining the microbiome with mental health as well. One of our panelists has a question about, are you thinking about, or are you considering the idea of performing a microbiome analysis from healthy people, like interactions with healthy people versus people with mental illnesses?
Well, it would be interesting to do as a clinical trial. And since most of the performances I'm doing are based on what I do, so it would be very difficult to get like ethics. I know that there was a questions about the ethical form. It's very difficult to get ethics certification to do these performances on someone else than me. So the only experiment I did with someone else was one I did with my partner, and it took me like 2 years to have ethics certification from my university because it involves not in simply interacting with others, but showcasing the results as an artwork. And if she wants to retract from it, then I'm going to have to destroy this artwork. So the ethics forms we need to do with the mothers also was quite tricky, because it was also involving babies, which were minors. And so that's why I'm usually, when I'm doing my artwork, only working on me and not working with other people.
Great. We have one more question if there's time for it. Yeah. Have you ever compared sort of the different sources of your data in the same city? So for example, your microbiome selfies from handshakes in one city versus the food in one city. Have you done any kind of integrative work there?
I can't hear you now. I'm going to shut down my video. It will be easier.
Okay.
Okay, he couldn't hear you well. So
yeah, can you repeat, because it says my internet connection is unstable. So
fair enough.
Just repeat the question, please.
Sure. Have you ever compared the different changes in your projects from the same city? So for example, comparing the microbiome of handshakes in one city to the different foods from a single place?
Usually I do only one performance at a time. So the comparing different cities for the handshake, that was to look at the dynamics of contamination in different parts of the world. But I'm not comparing food and handshakes and other stuff from the same city. So those are separate experiments.
Cool.
If that was a question.
Yep, absolutely. I believe it was. And one final question here. Can you describe a little bit of your process going from having all the sequencing data to turning it into the selfie? Like what's the process of going from data to art in that regard?
The process is quite simple, because I'm using an open platform called Gephi, which is a tool, a data visualization tool to look at your Facebook profile. So
Oh.
if you want to generate a network of your Facebook friends or your Twitter friends, then you can simply download this open platform, and instead of using my data from my social networks, I'm using sequencing data. And therefore, this is where you see these different clusters. But instead of having a cluster, the family, my friends, my colleagues, I have like different families of bacteria.
Cool. Very interesting. Thank you.
Thank you.
All right. Thank you for these also really great questions from everyone. Those were also very thought-provoking and gave us extra insights. So thank you again, François-Joseph, and thank you to all of our wonderful presenters today. You know what François-Joseph showed and what our Convergence course also demonstrates is that art and science have much more in common than not. You know, it requires a curiosity about the natural world, you know, be it how the brain works, or, you know, how the electromagnetic spectrum works, or what makes us human. We're curious and we engage in research practice, both in art and in science. And our students have really understood that. I think today's Science Symposium demonstrates that. And unfortunately, yesterday's crit session where all these art science teams presented was just, you know, in camera. It was not open to the public, but, you know, they were also like absolutely wonderful. And I find that the students don't just create pretty illustrations, which is what a lot of people think of when they think of science communications. What we've seen today were meaningful reflections of research questions that, you know, elicited empathy and that were humorous and thought-provoking and turned abstract data, science data, into an experience that really communicated with us on a much deeper level. And you know, your projects, through your research, you created knowledge that you couldn't create any other way. There's knowledge that you can create in science, but there's a different kind of knowledge that you can only create with art. And so you were able to sort of do both and so step from one into the other and sort of balance it out and sort of create this beautiful amalgamation of art science research. There's an author that I liked. It's Arthur I. Miller. He wrote the book Colliding Worlds. And he says that the terms arts and scientists have become meaningless and that we've all become researchers. And I agree with him. And I would even say that we are researchers and creators, all of us. And you've, you know, created beautiful examples of that proposition.